welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here's what I bring to you all from the world of medicine. AI algorithm that detects brain abnormalities that could help cure epilepsy and is developed by a UCL-led team of international researchers. The Multi-Center Epilepsy Lesion Detection Project, or MELD, used over 1,000 patient MRI scans from 22 global epilepsy centers to develop the algorithm, which provides reports of where abnormalities are in cases of drug-resistant focal cortical dysplasia, or FCD, a leading cause of epilepsy. To develop the algorithm, the team quantified cortical features from the MRI scans, such as how thick or folded the cortex or brain surface was, and used around 3 lakh locations across the brain. Researchers then trained the algorithm on examples labeled by expert radiologists as either being a healthy brain or having FCD dependent on their patterns and features. The findings published in Brain found that overall the algorithm was able to detect the FCD in 67% of cases in the cohort. This is particularly important as if doctors can find the abnormality in the brain scan, then surgery to remove it can provide a cure. This study on FCD detection uses the largest MRI cohort of FCDs to date, meaning it is able to detect all types of FCD. The MELD FCD classifier tool can be run on any patient with a suspicion of having an FCD who is over the age of 3 years and has an MRI scan. Smart contact lenses for cancer diagnostics and screening. Because of multiple capabilities, there has been much interest in using exosomes for cancer diagnosis and prognosis or treatment prediction. However, this has been hampered by the difficulty in isolating exosomes in sufficient quantity and purity for this purpose. Scientists from the Terasaki Institute for Biomedical Innovation, or TIBI, have developed contact lens that can capture and detect exosomes. The lens was designed with microchambers bound to antibodies that can capture exosomes found in tears. This antibody conjugated signaling microchamber contact lens, or ACSMCL, can be stained for detection with nanoparticle tag specific antibodies for selective visualization. This offers a potential platform for cancer pre-screening and a supportive diagnostic tool that is easy, rapid, sensitive, cost-effective, and non-invasive. In an initial validation experiment, the ACSMCL was tested against exosomes secreted into supernatants from 10 different tissue and cancer cell lines. The ability to capture and detect exosomes was validated by the spectroscopic shifts observed in all the test samples in comparison with the negative controls. Similar results were obtained when the ACSMCL was tested against 10 different tear samples collected from volunteers. In final experiments, exosomes and supernatants collected from 3 different cell lines with different surface marker expressions were tested against the ACSMCL along with different combinations of marker-specific detection antibodies. The resultant patterns of detection and non-detection of exosomes from all 3 different cell lines were as expected, thus validating the ACSMCL's ability to accurately capture and detect exosomes with different surface markers. Last study which confirms that COVID-19 mRNA vaccines are safe in pregnancy. The new study from the Canadian National Vaccine Safety, or CANVAS network, looked at data from participants across 7 Canadian provinces and territories between December 2020 and November 2021. All vaccinated participants were asked to self-report any health events during the 7 days following each dose of COVID-19 vaccine. The unvaccinated pregnant control group was asked to record any health problems over the 7 days before they filled out the survey. In total, 191,360 women aged 15 to 49 years with known pregnancy status completed the first dose survey and 94,937 completed the second dose survey. The researchers found that 4%, or 226 out of 5,597 of mRNA vaccinated pregnant females reported a significant health event within 7 days after dose 1 of an mRNA vaccine and 7.3% after dose 2. The most common significant health events after dose 2 in pregnant females were a general feeling of being unwell, headache or migraine, and respiratory tract infection. In comparison, 3.2% of pregnant unvaccinated participants reported similar events in the 7 days prior to the survey completion. In the vaccinated non-pregnant control group, 6.3% reported a significant health event in the week after dose 1 and 11.3% after dose 2. The That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.